Hi, everybody. Thank you very much.
Something that is
Okay. Right. So, yeah, I am Dr. Beric Gilbert. I am a lecturer at the University of the West of Scotland, and my background and area of specialization is looking at sort of the environment, the interactions between environments and parasites. I specifically focus on ectoparasites. So basically, before we sort of dive into the whole concept and everything, I kind of thought, well, maybe to give an overview and a context of where it all sort of began with bioindicators and that kind of thing. And it largely has to do with the effects that people or human activities have had on the environment, and in terms of the rising levels of pollution, which we've seen they've risen exponentially and that kind of thing. So most of this can be related back to the different industrial revolutions that have taken place, where we've seen changes in innovations and the resources that are used in order to develop the technologies that we have today. And along with this, when these changes have taken place, there's obviously been an increase in the amount of pollution that is produced and released into the environment, as well as a change in the types of pollutants that are released into the environment. And this has had an effect as well on increasing the pressure that's placed on natural resources. So the effects of pollution are not only because of the materials that are used, but it's because of the byproducts that essentially are released from the different processes that we use these materials for, essentially. And the problem with this is largely got to do, in terms of organisms, has to do with the fact that the changes that have taken place and the rate at which these changes take place are far greater than what many organisms, particularly sensitive organisms, are able to adapt to the changes. And this is a concept that, I mean, it's been published obviously in scientific literature, but it also is something that has popped up in popular science literature as well, such as the really well-known book, The Silent Spring by Rachel Carson. So for aquatic ecosystems, these ecosystems are some of the most severely impacted ecosystems globally, and that's because they are a repository for many pollutants that are released from human activities and, well, human and natural activities. And therefore, there's a need, because of their importance in sustaining life, there is obviously a need to monitor the health and the sustainability of these ecosystems. And one way of doing that is obviously using bioindicators. So bioindicators in the aquatic ecosystems, there are a number of organisms that have been used for this particular purpose. But the main overall goal of using them is obviously to delineate and determine the suitability of these ecosystems to meet the different services from an environmental aspect, so in terms of their functionality, but also right the way through to human uses and the reliance of people on these ecosystems. So in terms of the organisms that have been used, there's a wide range of species that have been used, from fish through amphibians as well through to bivalves. And then obviously most recently, and where I'm particularly focused on, is the use of parasites. So at this point, maybe you're kind of asking yourselves, but why on earth parasites if we already have all of these other organisms? Why are we interested in parasites? Well, firstly, I think it's got to do with the fact that parasites are a really diverse group of organisms in terms of from their morphology right the way through to the environmental sensitivity. And as such, there has been a growing interest in trying to understand these organisms in terms of the ecosystems that we find them in. So in natural environments, parasitism as a life strategy is ubiquitous and widespread. I mean, it's not just higher organisms that are infected by parasites. There are many examples that have been published where parasites have been found to infect other parasites. So we have this hyperparasitism that occurs in nature. And with this, there has been a growing recognition of the importance of these organisms within food webs and ecosystems and their functionality within these systems. So aquatic organisms, including parasites, are obviously exposed to a variety of stresses, and these stresses, the effects of these stresses obviously change under different environmental parameters, and this ultimately leads to eliciting different responses within organisms that occur outside of the normal range, and that's essentially what is monitored in this approach. So for parasites, where it all started, well, it's a concept that has been around for a while, but it's really kind of taken off in the last 33 years, roughly. And this has largely been due to growing interest in understanding the interactions between parasites and the environment. So looking at the interrelationship between the parasites and the macroenvironment. And some of the topics that have been covered in studies is looking at interactions and the effects of different pollutants toward different parasites, the changes in functional diversity of parasites in terms of once they're exposed to pollution, how do the faunal changes and that sort of thing relate to pollution levels. And then obviously the accumulation of pollutants and some of the physiological aspects linked to this, both within the host and the parasite specifically. So based on the effects that have been observed in parasites, two groups can be identified in terms of their indicator usage or usefulness as indicators, and these are effect indicators and accumulation indicators, where effect indicators are pretty useful in terms of providing information about the environment that they're found in, and this is largely based on the presence or absence of these organisms. And this is something that's been tested and explored using both ectoparasites and endoparasites. And then in terms of accumulation indicators, sorry, this has to do with the ability of these organisms to essentially accumulate pollutants in their tissues. And what has been shown, and you can see on the right side here on the graphs, is that, and particularly for metals, is that in some instances, certain parasite taxa are able to accumulate metals at much greater concentrations than what is found in the tissues of their host, and even at much lower exposure concentrations, they're able to essentially magnify the levels. This is something that has been shown mostly for endoparasites and not so much the ectoparasites. And some of the reasons around this has to do with the small body size of the ectoparasites and the low infection intensities that we generally find with this group of organisms or group of parasites rather in polluted environments where we don't necessarily find enough individuals and therefore there's insufficient material for the sort of more traditional chemical analyses that we would obviously do with this. However, for the endoparasites, there have been differences in the accumulation abilities that have been explored and tested, and overall, the acanthocephalans as a group show a really remarkable ability to accumulate and concentrate a wide range of toxic elements within their tissues. So why ectoparasites then? If these endoparasites are really good indicators, they have this remarkable ability to accumulate and bioconcentrate pollutants like metals in their tissues, and they occur at high enough intensities to provide sufficient material, why would we now be interested in ectoparasites? Well, the crux of it is really, in order to collect endoparasites, you've got to unfortunately kill the host. To do so. And the point of trying to use these organisms as bioindicators not only is to obviously look at the environment, but it's also to try and mitigate the use or the need of sacrificing the host in order to collect the parasites. So this is where ectoparasites essentially have the upper hand on endoparasites, and that's because they're found mostly on the outer body surfaces or the body surfaces of the host that are exposed to the macroenvironment, and they can mostly be easily removed for analysis without the need of sacrificing the host. So this is something that's really kind of important when you consider the sort of conservation status of hosts, of whether they're protected or not, essentially. So in terms of multiple stressors and looking at using these organisms as multiple stressors, in literature, it has been considered quite thoroughly about the from the host perspective, at least, in this regard. And that, when we look at parasites, has to do with the fact that the parasite and the pollutant are both stressors on the host, and therefore the host is naturally exposed to a wide variety of natural stressors. When parasites have been investigated and included in studies using model host organisms, what has been found is that infection by these organisms ultimately leads to a change in aspects related to the pollution tolerance, the oxidative stress, and biomarker responses in these model organisms. So ultimately changing the responses the host provides and is therefore not accounted for if you don't consider the parasite. But then if we obviously focus now on the ectoparasites and consider them, there are some studies that have looked at this from both the field and laboratory aspects and under different scenarios of stressor-host-parasite associations. So for today, I'd like to focus on one group of ectoparasites, just to sort of draw it in as a group of model organisms or proposed model organisms, and that is the monogenians, the Gyrodactylidae. So these organisms they infect a wide range of aquatic organisms, which include fish and amphibians. One of the main sort of unique features of the Gyrodactylids is the fascinating reproductive strategy, which they've sort of been synonymized as sort of the Russian dolls. And as you can see, is highlighted on the diagram and the micrograph, that's the uterus of the parasite. And inside the uterus, you can find several generations of the parasite in one individual. So in this case, the diagram shows that there are 4 different generations. So the embryo has its own embryo, which has its own embryo, essentially. And it's this reproductive strategy that under different environmental conditions, which include stress conditions, are in a way favorable to the parasite because it means that the parasite can essentially exploit the environment quite rapidly by expanding its population relatively rapidly. And this is something that obviously is a major consideration when you sort of consider the aspects around aquaculture and that kind of thing. In the fact that these parasites can become problems very quickly. So from an ecotoxicology aspect, the group is one of the most sort of or more explored groups of parasites that have been studied and that kind of thing. And this is what you can see on the graph over here. The pink bars are the Gyrodactylids, and they've mostly been assessed for a wide range of types of pollution that they've been exposed to. Back in 2019, a paper published by Tadari et al. Suggested that the genus, a certain species of the genus Gyrodactylus could be used as model laboratory organisms for this purpose, and that's largely because of the fact that they have been extensively studied for a number of different aspects. Some of which include the ecology and the environmental aspects of these parasites. So specifically on pollutants, this is something I did back in published in 2021 as a postdoc back in South Africa. And what I tried to do was I was trying to look for a way that we could, try and identify the specific responses of these parasites, specifically monogenians, toward different types of pollutants. And I conducted a bit of a meta-analysis about this. And really and truly, even when you focus on the Gyrodactylids, what we sort of found at the end of the day was it's really a mixed bag of responses. Some literature says that the parasites are sensitive to a particular pollutant, and in other cases, for the same type of pollutant, they'll say that they're not sensitive, for instance. And that's what you can kind of see on the graph, or on the table here, at least, relating to the Gyrodactylids. If you look at the first column, over here is the effect sizes of the effect of the pollutant on the intensity of the parasite. And only really for metals, the category which we call disturbance, and this was really because of it was a category identified because, like in literature, the studies hadn't really identified a specific type of pollutant. It was just that the environment was affected by that pollutant. So the disturbance category and then exposure to crude oil, they really provided or gave the highest effect sizes in terms of the pollutant categories that we had assessed. And perhaps part of this fuzziness that we find around the responses and the results for this and the mixed responses could be because of the mixed effects associated with multiple stressors and that sort of thing going on toward these parasites. So again, from the multiple stressors aspect, this is something that has been explored under laboratory conditions for some taxa. One study in particular back in 2019 is by Soleng and colleagues, 1999, sorry, back by Soleng and colleagues, where they looked at the effects of aluminium under different conditions where they manipulated pH of the environment toward Gyrodactylus salaris, which infected the Atlantic salmon. So what they did in their study was they used various concentrations of aluminium at 3 different pHs. And what they found was that when the parasites were exposed to only changes in pH, there was very poor effects of the alkalinity and acidity of the medium toward the parasite. The metal alone did impose toxic effects, particularly at high concentrations, which were demonstrated as a reduction in the intensity of the parasite. But then what they did find was that there was an additive effect of the metal toxicity being enhanced by low pH. So particularly, and that's what you can see in graph number C over here, is when the parasite was essentially exposed to very high pH, or to low pH, sorry, the metal toxicity increased exponentially and reduced the amount of time taken for the population to become reduced, obviously was shortened. So in terms of, so that's for a specific pollutant and the environment. And this is the multiple stresses aspect in this case has also been explored by looking at the pollutant and the host's effect toward the parasite. And this has been done with a couple studies, particularly focusing most recently on microplastics and the plastic polymers that perhaps you find in the environment, as well as nanoparticles of metals. And overall, what was essentially found by all of these studies is this, regardless of the pollutant that the parasites were essentially being exposed to, once exposure had taken place of parasites that became detached or were removed from the host, the toxicity of these substances all of a sudden started to increase towards the parasite versus if they were attached to the host. And these results essentially imply some form of host-mediated or host-related factors that essentially reduce the toxicity and potentially the bioavailability of these toxins, or toxicants at least, toward the parasite. And in a way almost protecting the parasite. So it's another sort of level within the whole multiple stresses idea and concept around parasites. So basically, just to wrap things up, so to conclude, in terms of the whole idea and the approach of multiple stresses and ectoparasites, there are obviously some examples that we can compare the effects of multiple stresses and the aspect of it with parasites, but generally we really do find that there's a real mixture of these effects, and this is likely dependent on the different conditions where these studies have probably taken place. And overall, the mixed effects really make it quite difficult to draw any conclusions about the interactions and the dependencies of pollutants and their effects towards these organisms. And it is really largely dependent on the pollutant-host parasite association and relationship, as well as the different environmental factors that would essentially modulate the harmful effects of these pollutants. However, based on the responses that have been documented so far, and specifically for the aspect of bioindicators, using these organisms as bioindicators, these parasites definitely do have, or ectoparasites definitely do have potential as bioindicators. And this is largely due to their sensitivity towards changes in the environment. So they can add to the environmental monitoring approaches and that kind of thing that we currently use. And this is particularly with regard to the early detection of pollutants in the environment, as these organisms are more sensitive than the host in some aspects to lower exposure concentrations. Thank you very much for listening.